Would you like me to start
Ye
or just
yeah,
give it?
if you want to give like an intro to
Okay.
yourself. Yeah.
Yeah, so I like Brianna said, I'm Brad Slaughter, a senior botanist with Orbis Environmental. I'm starting to I'm like thinking about how long I've been in this field now. I'm like, ooh, it's been 20 years. So I'm starting to feel it. But so I've been working with Orbis since 2017 as a botanist, and I was senior botanist. Prior to that, I was an ecologist and later botanist with Michigan Natural Features Inventory in Michigan. Primarily still work in Michigan and in Indiana, doing all sorts of things, fun things, not so fun things. For like probably like most of us. Um, so today we're going to cover, initially I think the idea for this brief workshop here was to cover all of the biomes of Michigan, but there are 11 species, and so rather than trying to cover one species every 2 minutes or less, there's really kind of a lot of terminology and imagery to look at here, and so we're only going to focus actually today on four species. If you are familiar with the Michigan flora, either the book or online, this is the first four species you see in the key. And so what we're seeing here in this cover slide is Bidens cernua, which is one of the species we'll be covering here today. Um, it's interesting looking at different resources. It seems like there's quite a bit of taxonomic uncertainty around Bidens, especially as far as the species richness goes. I find references of anywhere from 150 to 280 species worldwide, which seems kind of like a bizarre range. But again, there is some taxonomic uncertainty. 25 of these are covered in the flora of North America. We have 11 species in Michigan. 10 of those are on Michigan Flora Online. There is one species we have that I just don't think has been collected yet, so it's not reflected in the flora. Biden's, Bi is two and dens is tooth, so that refers to the on fruits. That's where the name comes from, and they are principally wetland species. And so eight of the 11 species we have in Michigan have wetness coefficients of less than zero. So they are of particular interest to wetland delineators and people doing wetland regulation. Um, so there's kind of a lot to look at with the flowers, and this is why I want to only cover the four species today, is because it really pays to kind of look at this diagram. And I hope I know on my screen it looks like I've got some stuff blocking it, and maybe less so there. Um, and I did borrow liberally from online sources for the imagery and so on. So if you see anything of yours, I hope I either put your name on it or but appreciate using it here for this the purposes of this little workshop. So this diagram, it's just got a cross section of a typical flower of the family Asteraceae. So you know that the flower, I said I already made that mistake. It's a flower head, so it's composed of many individual flowers that are arranged in what most people would look at and call a flower. So A is a cross section of the inflorescence. The showy yellow ray flowers that are characteristic of Bidens are indicated with the lowercase r. The disc flowers D, so the disc flowers are the ones that kind of are in the center. The involucre is something that you'll see referenced when you're keying out Asteraceae in general. So that's that series of bracts that sort of subtend the individual flowers. So that's indicated with the IN. If you look at this zoomed in close-up diagram that's labeled with a capital B, that is a disc flower. It's closed in that case. If you look at D, it's open. You can see the little lobes of the corolla, so it does look like a tiny little showy flower. Wouldn't look like much if you look at it without a scope or a hand lens. And you can see that a couple key features here. Um, B, so the lowercase b here. So at the base of each of these individual disc flowers, there's a scale that is referred to, you'll see in different keys as the chafe or as a palea, not to be confused with a palea in grass flowers. So there is a lot of botanical terminology that is used maybe in different ways for different parts of the flower and so on. So usually you'll see this reference as the chafe. It can be fairly colorful. The bristles you see are actually the modified calyx or sepals. And in Bidens, they're modified into these barbs or these bristles or awns that have barbs on them. And so if you look at that, you can see that already in the you know the the pappus here is what that's called, the calyx that's modified in that way. If you look over at capital letter F on the right, that is the fruit. The fruit of Biden's is you'll see it referenced either as an achene or as a cypsela. The apparently the critical difference or distinguishment between those two is that an achene is born from a superior ovary and a cypsela is born from an inferior ovary. So you have to go way back to your initial botany classes, but a lot of references just don't bother making the distinction and just refer to it as an achene. And you can see that it has that pappus is retained there, and those bristly awns are retained, which are that diagnostic thing about Biden's. So the key, we're just going to run through the first part of the key from Michigan Flora Online and the book Michigan Flora. And we're focusing on the species that are indicated here in the green glowing font. So we have one aquatic species, like true aquatic species, and then we have several species with leaves below the inflorescence that are simple. We're going to focus on those today and leave the compound leaf species for next time. So when we go to that very first line in the key, it states leaves mostly aquatic and fully dissected. We have that one species, Biden's Beckii, which honestly, I'm not sure that I've seen this or recognized it in the field. Maybe not just not doing a lot of aquatic botany work. I just haven't really come across it or looked for it. But you could see this is, our true aquatic Bidens that occurs in shallow lakes, ponds, streams, and so on. And if you look at just the flower heads here, there's nothing much to really distinguish it there. It looks like any other showy Bidens. But when we take a look at the leaves, um, they're very different. So you can see these really deeply dissected leaves, and they look like they're whorled. If you take a look at the image on the right here, it almost looks like there are four leaves. There are actually two leaves that are opposite. Um, so the leaves in general in Biden's are opposite, and in this case, these are also opposite, but they're branched really close to the base of the stem here, and so it looks like there are four whorled leaves instead of two, really divided um, opposite leaves. And so you want to make sure to look for those. You have to kind of go under the water to look for those. If you find those, then it's pretty obvious what this species is. If you look at the upper leaves, you can see that once the plant emerges above water, it develops simple leaves that are toothed, but it can be confused with other Bidens. And you will often see the next Biden species that will cover in similar kind of shallow water situations. And some people might just look at it and not look underneath the water and confuse it for this species. So you really kind of want to make sure we have these really dissected leaves. Another thing to look for if you can find it, later in the season when it's fruiting is this species, if we refer back to that cypsela or fruit, and those pappus awns, the bristles at the ends of the fruit here, they're really wild in this species. They can get to over 2 centimeters long, and they're really kind of spreading and very distinctive compared to everything else that we're going to see here. So Biden's Beckii, aquatic, big lawn awns on the cypsela, deeply dissected aquatic leaves. So very distinctive, occurs statewide, not particularly common. Like I said, I honestly don't remember if I've seen it before, but it's a neat plant. When we go to the next step in the key with simple leaf plants, you've got two options here. The first one's the easiest, the leaves being sessile, and some are connate. So connate leaves are more or less connected, so it's like they clasp almost. And this species typically, or almost always, has showy rays that significantly exceed the head here is Biden cernua. So Biden cernua is the nodding beggar ticks. And most of you are probably familiar with this species. It is definitely one of the most common and also one of the most noticeable because it forms great masses. It's very showy. Any wetland situation where the water level drops over the course of the summer, mud flats, places like that, this thing can occur in abundance. You see that on the left with the showy rays. On the right, you can see how those stem leaves there, the simple stem leaves, are somewhat clasping, or they're somewhat connate, not really clasping, I guess, but the connate, so they just kind of connect more or less at the base of the stem. So you want to look for that. Um, Also, so this species, in you won't find this, not every single head is like this, but the reason they call it nodding beggar ticks is if you look at that central image there, you can see that those heads do at maturity tend to nod and sort of, like a sunflower almost, where they're oriented vertically, and that that is distinctive later on in the season. You can see the cypsela on the right. There's nothing in today's talk that really is worth focusing on with that, but that is kind of the general structure of most of the fruits that we're going to be seeing with Biden's. So you can see again, it's got those bristles. Even though Biden's is named, bias two, a lot of our species have more than two bristles. So quadens, not sure what we call that, but that is the fruit there. On this species, it tends to be kind of plump. Um, Again, that's probably one of the easier species to identify based on those sessile leaves in the big rays. And you do want to make sure to just kind of look down the stem because um, the next couplet in the key, there are two species that have leaves that are mostly narrow to distinct petioles. So it's kind of one of those, those relative characters that can be a little bit troublesome if you don't have something in comparison. But these species also typically don't have rays, or if they do, they're not showy. And these two species, I sometimes still turn these in and put the wrong name on them. It's annoying. There is some careful, this is where reading a key carefully, unlike what I do sometimes, is helpful in identifying these. And we've got Biden's camosa as the first species. Um, so you can see on the left, these are the Bidens that they can be very abundant, just like Biden's cernua, but they're the non-showy ones. They can be large, robust plants, but without heads, they just kind of look gangly, and they can form immense stands. But you can see really clearly here on both of these images, that the leaves, are definitely distinctly narrowed to that petiole. So you don't have those connate leaves, or leaves that are almost touching on these species. And again, you don't see the rays here, or if you do, they're very small and not showy. There are no rays, right? Listen to me, Ray. If you know the reference, you get an extra credit for something. But these are the flower heads here of Biden's camosa. So again, there's just nothing there with the rays really. Oh, I should go back here. I know how to go back. Yeah. So another thing I want to point out here is that if you look at the both of these images, really, you'll see that there is not that tendency for those flower heads to sort of um, nod. You can see like maybe a little bend here in the picture in the right, but they don't have that nodding tendency like Biden Cernua does. Now, this is a part of the key where you really have to look. Really closely at these cypselae or achenes because some of the characters that we're using to identify these are pretty subtle. In this case, the cypsela of Biden's camosa is said to be, it's really flattened. So it's compressed looking, looks flat, and it's going to tend to have three awns. So two long awns on the margins, and then one shorter awn associated with a midrib, of which there's usually just one distinctive or somewhat distinctive midrib. So only on one of the sides of this cypsela are you going to find that. There is a note in the key, and this is, I think, where sometimes being sloppy and just grabbing some quick achenes out of a head and looking at them isn't sometimes doesn't cut it because the key does ask that you sample from the center of the heads. And so you want to make sure to kind of get a representative sample of the cypselae from that portion of the inflorescence it's asking about. In this case, again, you will find cypselae for Biden's camosa that have two awns or four awns sometimes, but the general pattern is to have those three. The other thing about this species is you want to look at the base of the cypsela because these little bristles that occur along the fruits are retorse or retorse, meaning they are bent backwards, so they're pointed down all the way down the sides of these cypselae. So that's what the arrow is pointing. If you looked at that under a microscope or with a hand lens, look at the very base of the fruit to make sure that those are still pointing down. It's a critical difference with this species and the next species we'll look at. Here's another image of a cross section of the flower head of Biden's camosa. Again, you can see those individual cypselae there. They're very flat. You can't really count the bristles too much there, but you just kind of get a sense for the overall structure um, of this species. The other species that it can be confused with, again, has simple leaves that are narrow to distinct petioles, just like it, is Biden's Canada. And so I didn't really mention the range or put maps on. So Biden's Camosa, if you look at Michigan Flora online, you'll see that most of the records are in the southern part of the lower peninsula, maybe midway into the south. I've collected it in Traverse City. There is an old record in the upper peninsula. Biden's conida, what we have here is this tends to be the more common species for those of us in northern Michigan. So I don't necessarily see these together all that often to be able to compare them like in the field. But range wise, if um, we're going to tend to see more conida up here and down south, more of a mix. But again, just so we look at the petioles here. Otherwise, the form of the plant looks very similar to that of Biden's camosa. But take a look at the fruits here, are quite a bit different. So again, looking at the cypselae from the center of those heads is important. So we want to get those. Hopefully, we have mature ones. You can look at it under a scope, the hand lens, preferably a scope, because you really want to get pretty good magnification on these. And you can see that as opposed to the cypselae of Biden's camosa, we're looking at something here that tends to have four awns. So this species, the fruits tend to have two distinct midribs. So you've got the edges. So the big lawn awns are on the edges, and then you have two short awns associated with a midrib on each side of that cypsela. Also, these cypselae are said to be warty. I don't think that the warts are absent per se from Biden's camosa, which we just looked at, but they're more distinctive typically in this species. As I mentioned earlier, we were looking at the little barbs on the fruits of Biden's camosa because we were looking at the base to make sure that those barbs pointed down. Reason being, in Biden's Canada, those barbs at the base of the fruit, as indicated by those arrows, and I apologize, it's kind of blurry, but that's kind of the best image I had here. You can see that those are actually pointed up. So those are anthers. Instead of retrors. And you want to find, they can be sparse, so you don't see many of them. But you do want to look at that base of that fruit to make sure that the orientation of the barbs at the base. That's going to be critical to identifying these. And I think there's a form of this species. So the other thing is not to get confused by what the barbs are doing in the upper parts of the fruit, because they can vary. You can have anthers. Usually the barbs are retrors on the upper part of the fruits of the species, but they can be anthers. So beware. Make sure to look at the ones down by the base. There is another distinction, sort of a more of a field character that people use. And again, I think I've not seen these in abundance near each other enough to really feel confident employing this in the field. But if you look at the flower heads of both species, Biden's camosa on the left, Biden's Canada on the right, there are some differences. And they have to do with the on the left, and you can't really see it that well here, but the anthers on Biden's camosa are more or less they're contained kind of within those corolla lobes. So you don't really see them sticking out significantly. On the right in Biden's Canada, the anthers, are clearly exceeding the corolla lobes. The color of the chafe is also said to be different. So the chafe being sorry, the corolla lobes themselves, I think, are also different. So the coloration here, and I don't know how well, this holds up, but it's said that the corolla lobes in Biden's camosa are more of a pale yellow, and they're orangish in Biden's Canada. So a little bit more showy in Biden's Canada. I've kind of looked at this, and I think sometimes I forget what the distinction is in the field, so maybe that's why I don't notice this as much. These images, it looks quite a bit different. So they're not quite at the same stage of maturity here, but that is a field character that you can try to use to separate Biden's camosa on the left. So camosa inserted anthers, pale. Biden's connata on the right, exserted anthers, orangish or more showy. Another thing to look at, and again, this is one of those things where you might look at these and say, "Oh, it doesn't look that much different," but the filaries, so those outer bracts on the that are kind of at the base of the flower heads and Biden camosa are typically more numerous and they're more foliaceous, so they're more leaf-like. So that yellow arrow on the left there is pointing to, you can see that there are some of these inner filaries that are shorter and stubbier, but it has these big leaf-like filaries as well on the outside. You can see that they're much longer than the disc, so it just looks busier in general. On the right, Biden's connata is going to tend to have fewer of those outer filaries, and they tend to be more linear. So not so busy looking, not as leaf-like. Again, that's one of those characters. I think this is a pretty strong thing when you look at a number of flower heads. It should be pretty obvious. If you look at an individual flower head, sometimes they might, equivocate over whether it really looks that much different or not. But that is something to look at as well in the field. Again, just looking at those fruits together. So on the left, you have Biden's camosa. Um, the cypsela Biden's camosa being flattened, that most one somewhat distinct midrib, three awns, and you can't really see the that on the. I guess you can a little bit see that those barbs, at the base of the fruit there are still retorse. On the right, you've got Biden's connata, which is said to be flattened, but not as flat, maybe. So they're like a little bit squished. But it has four awns. It has more those two distinct midribs, and you can't see the barbs at the base of this one. But remember that the barbs at the base are actually anthers. They're pointing in the opposite direction in this image than the ones that you can see clearly up at the top here on the awns. One thing I should mention, so we're just looking at the species with simple leaves today. We're not looking at species with compound leaves, but of course, nothing's that simple. You want to make sure to look at the leaves when you're in the key and trying to make this distinction. Look at the upper leaves and the central leaves on the stem. If you look at the lower leaves, sometimes you'll find that the lower leaves have lobes. They're not fully compound, so especially on the right there, you can see that there's clearly still laminal tissue. Um, it's not divided into a distinct leaflet on the left. It looks a little more so like it's divided. So you will see some of that on the lower leaves for these two species that are otherwise said to have simple leaves. So just make sure you're looking as the key tells you to in the on the upper and middle portions of the stem to make that call. Nothing's that easy. I don't know how this was kind of intended to be a little bit interactive. Just a couple of these uh, images just to see what you think if you want to make a call on what they might be. There's the first one. We probably don't have a way to do this, but what I was going to point out here is this is a little bit different in that you don't see the really distinct petiole up here on this plant, but it still does narrow, and you can tell that those flower heads, there are no rays again. So the combination of narrowed leaves, no rays, and look at those foliaceous outer filaries. So look how leaf-like those filaries are. So this would be an example of Biden's camosa. As we looked at earlier, this one right away, you can't see the leaves very well, but you might be able to tell that they're connate. You can see that the flower heads are nodding. You can see the showy rays. So we have Biden's cernua here. And this one, and I see a hands. I'm not sure if there's a question but this is you can see that these are very flattened achenes. It's just an image from a different source here. Flattened achenes are cypselae. You can see how sometimes I'm not seeing three awns here. I'm just seeing two. So I'm not sure where these were from in the flower, but of all the species that we saw today, you could still make the call that this is going to be Biden's camosa. Another image of Biden's connata fruits just to see the difference. Again, you can see how they're variable. I think what I showed earlier, it's too easy to look at what the character is and show you an image where they all have the nice four awns. In this case, it could be that one of or two broke off here, but you can see that the fruits are warty. You can kind of see if you look closely, you can see that there are ant force barbs at the base of the fruits here. So these are going to be Biden's Conata. And this was my trick question if we did the quiz when people tried to answer, and then I could say, "Ha, we didn't cover this yet." So this is one of the compound leaf species that we'll cover the next time. And so with that, I guess I'll just open this up for any questions. Thank you.
Yeah, thank you so much. We definitely enjoyed the quiz at the end, and I'm gonna try to figure out a way, if like when we do the second half of this Biden's presentation, maybe we can figure out a better
It
interactive.
would be great to have that interactive so
Yeah.
we could talk through some of the, yeah.
Yeah, that was great. I'm sure everybody appreciated it. We do have one question. It says, is the midrib present on both sides of the seed or just one?
For Biden's Camosa, it's on one. On Biden's Canada, it's on both. And so you'll see that that midrib is usually associated with a short on. And so sometimes the on will help point to where it is because if you look at a fruit and you're like, I don't see a midrib. If you look at the on and see it's coming on the other side, flip it
Mhm.
over, take a look, you'll see the midrib.
Gotcha. Okay.
In fact, I think the image on Michigan Flora online, there's an image of a Cypsela for Biden's Camosa where you do see that third on, but it's coming off the bottom, so you don't, there isn't a distinctive midrib on the surface of the image, so it can be confusing, but it's because it's on the other side.
Yeah, I can see that. Yeah, I want to see if there's any other question. I think that's all for now, but yeah, we really appreciate your time and we look forward to the next presentation. And yeah, we're hoping this is kind of our trial for the mini series, so it is a little bit fast paced, but we really appreciate the detail and care you have with each of these species today. So thank you so much, and we'll be in touch.
Yeah, thanks for having me.
Bye, everyone. Thank you again.